Regulation of blood pressure. Blood pressure, single most important factor regulating the cardiovascular system. Mechanisms can be divided into short term, intermediate term, and long term. These mechanisms are for the regulation of blood pressure. Coming to short term regulation, it is card central nervous system controlled. It is rapid acting. They are baroreflex, chemoreflex, and central nervous system ischemic response. Coming to intermediate term, uh, regulation of blood pressure, which is activated within hours by vasodilation and vasoconstriction, and it has mechanisms like capillary fluid shift, stress relaxation, renin angiotensin, aldosterone system. Short term is RAS. Coming to long-term regulation of blood pressure, activation within days, and regulation is by kidneys, pressure diuresis, co-regulates fluid volume and urine output. Short-term regulation, baroreflex. Increased blood pressure via baroreflex causes negative feedback and decrease in blood pressure. Make- so the stimulus here is increase in blood pressure. Receptor, baroreceptor, they sense distension. Afferent, baroreceptors are present in carotid and aortic. Uh, Carotid baroreceptor, aortic baroreceptor. The location is drawn here. You can see the arch of aorta, aortic arch receptor, and carotid sinus, which is small dilatation in internal carotid artery. So afferent from carotid is cranial nerve 9, hearing nerve, and from aorta is cranial nerve 10, Lyon's nerve, which goes to nucleus tractus solitarius in medulla. From there, glutamate is released, and efferent glutamate is sent to caudal ventrolateral medulla, CVLM, and GABA is released, sent to rostral ventrolateral medulla, which is sent to sympathetic inhibition to heart and blood vessels. On the other hand, glutamate is sent to nucleus ambiguous, which is sent to parasympathetic activation, which is acting on heart and blood vessels. Response is decreased blood pressure and decreased heart rate. Note Mary's law. Increased blood pressure leads to reflex decrease in heart rate. That is, heart rate is inversely proportional to blood pressure. Experiments. Occlusion of common carotid artery or clamping below the carotid sinus in internal carotid artery. Decreased blood flow to internal carotid artery. Decreased pressure. Baroreflex is inhibited. Increased blood pressure, increased heart rate, sympathetic activation, clamping above carotid sinus, increased pressure, baroreflex activated, decreased blood pressure and decreased heart rate, increased sympathetic inhibition. Clinical aspects: supine position on standing immediately, decreased blood pressure, baroreflex inhibited. If CNS underactivity, fall due to postural hypotension. If sympathetic nervous system activated, release of norepinephrine will increase the heart rate, will increase vasoconstriction via alpha receptors, and increase total peripheral resistance. You can imagine alpha constricting an artery because alpha receptors are vasoconstrictors. Carotid sinus massaging. Zermatt-Herring test. Carotid sinus massaging will activate baroreflex and will decrease the heart rate. So when you massage carotid sinus, it should decrease the heart rate. Used in treatment of paroxysmal supraventricular tachyarrhythmia, PSVT. Carotid sinus massaging is used in the treatment of paroxysmal supraventricular tachyarrhythmias. Other baroreceptors, atrial and pulmonary artery baroreceptors, low pressure receptors, volume sensing receptors. Two types of atrial stretch receptors are present, type A and type B. Type A is activated during atrial systole. Type B is activated during atrial diastole. So there are two types of atrial stretch receptors, type A during systole, type B during diastole. Regulation at different levels for of blood pressure. Baroreceptor is the first line to regulate blood pressure. Stimulus means mean arterial pressure of 70 to 140 mmHg. Response up to mean arterial pressure of 180 mmHg. Chemoreceptor, second line for blood pressure control. Stimulus is mean arterial pressure of 40 to 70 mgs. Um, Chemoreflex direct effect activates parasympathetic nervous system, decrease heart rate. Indirect effect activated sympathetic nervous system, increase heart rate. Effect on heart rate is variable. Central nervous system ischemic response. Last line of control, last ditch stand mechanism, stimulus, mean arterial pressure less than equal to 40 mmHg, response, decreased cerebral perfusion due to low perfusion pressure, activation of RVLM, um, sympathetic nervous system, and increased mean arterial pressure as high as 250 mmHg. RVLM is rostral ventrolateral medulla. Blood pressure waves, also known as Mayer waves and vasomotor waves, oscillation of nervous control mechanisms, Traube-Herring waves, fluctuation in blood pressure synchronized with respiration. During inspiration, decreased systolic blood pressure. During early part of expiration, increased arterial pressure. So during inspiration, systolic blood pressure is decreased, and during early part of expiration, arterial pressure is increased. Cardiac reflexes, Bisol Gerish reflex. So serotonin is released from injured tissue in the heart, activates Bisol Gerish reflex, bradycardia, decrease heart rate, and hypotension. So when heart is injured, heart rate and blood pressure is dropping. Cushing's reflex. Head injury will increased intracranial tension, will cause bradycardia, but that is reduced heart rate. Cushing reflex, head injury, increased intracranial tension, which in turn reduces heart rate, bradycardia. Brain bridge reflex. For example, fluid overload in congestive cardiac failure activates sinoatrial node, right atrium enlargement, tachycardia, increase heart rate. Brain bridge reflex. Fluid overload congestive cardiac failure activates sinoatrial node in right atrium enlargement and causes tachycardia, increased heart rate in fluid overload. Chemical regulation, vasoconstriction mechanism, increased blood pressure. For example, urotensin is most potent, norepinephrine alpha agonist, endothelin produced from endothelial cells of lungs, ETA receptor, ETB receptors, endothelin 1 overactivity, pulmonary artery hypertension. Treatment is bosentan, endothelin blocker, vasopressin, factor 6 receptor decrease blood flow. Sorry, it is factor V1 receptor. Um, so vasopressin is factor V1 receptor and decrease blood flow. Terlipressin used in treatment of esophageal varices, angiotensin II. Vasodilator mechanism decrease blood pressure used in treatment of hypertension. Example calcitonin G related peptide CGRP are most potent. Nitric oxide secondary messenger CGMP used in treatment of angina. Natriuretic peptide ANP, BNP, CNP, second messenger CGMP, nasiretide, BNP analog is nasiretide and bradykinin.